and this is the Sample Preparation Challenge discussion forum. So we're going to be talking a little bit about sample management, tracking preparation, and trying to identify areas where we have bottlenecks and areas that could be improved. So on the panel today, we have Rob Niesman from the Rabu University. We have Kurt Zymek from the Donald Danforth Center, and we have Maud Limou from the RFI. So if you'd like to introduce a little bit about yourself.
I'm Rob. I work at the Department of Microbiology, Faculty of Science at Oxford University. I've 6 years of experience in EM substance preparation. I mostly work for spectral microscopy, sometimes some practice or hyper-3D substitution. Also chemical preparation. Roughly anything you can find I find interesting or have tried or want to try.
Hi, everyone. I'm Kurt Zymek. I'm in St. Louis, Missouri at the Danforth Center, and it's a plant research center, but over the last decade or two, I've done a few other kinds of samples too. And I do both cryo and room temperature, array tomography, volume EM, FIBSEM, serial block face, you name it.
Hello, I'm Maud. I'm working at the Franklin across the road. My main interest in my work is on cryogenic workflows and how to link mesoscales to nanoscales. So focusing mostly on cryo because if we want to do subtomographic averaging and structural biology in cells or in tissue, we need to go cryo. Working a lot with X-rays, STEM, TEM, fluorescence, you name it.
And I'm Kirsty, and I work at the Abraham Institute, and I have potentially, in some people's eyes, devolved in my microscopy skills. I started off doing single particle cryo-EM and have gradually moved through cryo-sectioning, Tokayasu, to room temperature resin, and now I'm going back through free substitution. I've worked on a variety of samples. My most memorable ones have been virus structures of extreme files, right the way through to whole tissues from various animals, and now onto tiny little fiddly awkward worms. So I think we're going to maybe start asking the panel a question about what sample preparation strategy do you use when you're faced with a new specimen? So maybe if we start with Kirk, seeing as Rob's microphone is on the blink.
Okay. I guess there's two things. I would put them into the cryo workflows with free substitution, in which I think there was some nice presentations today along those lines. I mean, those protocols are all in the ballpark. I usually start with more metal than not my first round, to be honest with you. Sorry for the environmental folks on here. And then I work backwards, because the worst thing you can do is go through all this effort of weeks and have low contrast in your images. So I usually start and then I back out if I can. To be honest with you, since I work with plants, I've been using the Hua protocol. I think that was 2017. It basically was designed to do more uniform brain OTO protocol. And turns out that I've used it on a lot of different samples, and it's been pretty robust in my hands. So that's my tip to you. It's UA. I think it's 2017 Nature methods. I should know it since I'm citing them right now. But, okay, that's my starting point. And start with a lot of metal, and then I back off from there if I feel it's not worth it.
Thank you. Rob, do you have any preferred method?
First, I start out asking the people who give me the sample, what do you want out of this? Where do you want to go with the sample? And dependent on that, I can say, okay, let's go this way or that way. My standard approach for free substitution is often going to epoxy resin, and then I had, let's say, 2% of osmium, 0.2 UA, only 1% of water, because I don't have my UA in a methanol stock solution, and then continue. That tends to work. Their trick mostly is dependent on tissue. You should use different high pressure freezing approaches. And my favorite is actually going ultra low temperature embedding in HM23, because I really like the amount of contrast you get. This is for TEM then. You only have UA in there, and it gives you extremely nice contrast and gives you also the tiniest details on protein complexes.
So for cryo workflow, you see it's not simpler, but because we don't use staining, we don't invade, we don't dehydrate, we don't do anything like that. The focus is on the quality of vitrification. And for that, you have two techniques and two approach, and it's very sample dependent and the thickness of the samples. So you can go for a route of punch freezing. In this case, your sample should be thin if you want to have high quality vitrification. If you go beyond that, you have to go high pressure freezing. And this is now the moment that you enter into the realm of cryoprotection and cryoprotectant. There is a lot of work that has been done by multiple groups, but also you're going to listen to it a bit later on how to find the best one. And obviously the question for us is very simple, because we want to have the possibility to be fully compatible with cryo-electron tomography and septographic averaging again. So we want to make sure that we don't disrupt anything too much, so we can keep the structure of the protein completely intact. So they are mostly the two branches I'm going for.
Okay, thank you. So hearing that it seems that you all have a very specific starting point for your sample preparation protocol. Do you have preferred methods? So like Rob said, once you've spoken to the scientists, you decide what you want. Do you have preferred methods to go down for certain like organelle identification or for certain feature identification? Or do you always start with the same initial robust technique and then try and refine?
I think I start out with in the direction that they want to go. Because for me, there's no use in spending, let's say, 2 weeks on a free substitution if they're not interested in having high quality details on organelles or anything. If they say, okay, I want to identify this structure here and I want to do immunolabeling, then I often refer to Tokiyasu because it just works. You can say everything about contrast, but the method just works and it's still very robust in working. The minute they are interested in having more structural details with immunolabeling, for instance, then I would go for high pressure freezing free substitution, or even high pressure freezing free substitution rehydration, and then again Tokiyasu, also depending again on the sample and how well the antibody works.
So I should say to anyone in the audience, if you have any questions, please raise your hand.
Yeah, thank you. It's actually kind of a follow-on to something you just said, Rob. So in my experience of working with users who maybe aren't as familiar with volume EM techniques in general, they come to you and they say, I want X. I'm just wondering how frequently that X, that thing that they want, is actually what they want in the end, right? And the kind of the second point to that is, because you said that it's not worth going through the effort of like a free sub protocol for high resolution detail if that's not what they want. But if that turns into what they want, right, but they don't realize it until after you've gone through some other efforts, like, can you just talk about that as like an idea?
Okay, this is why I spend, let's say, at least 1.5 to 2 hours talking to the people for which I do the samples to figure out what they really want. And often it turns out we've seen this brilliant article, this very nice image using an SEM, and then look at the image and I said, no, this is TEM, this is not SEM, or vice versa, where they say, yeah, there's large overviews using TEM, and then it turns out it was SEM. So in often case, especially if you work for other people, it's a part in terms of education and really talking about getting also a bit background into what their final aim is with their research, where they want to go, what their plans are, to also see, okay, if I do it like this now, they'll probably come back and have at least 20 weeks more effort to put in it, or I can just put in the effort now a bit more, and then we're done in 3 weeks' time, and then everybody's happy in the end. So it's really expectation management and communication. And I'm not that great in communication, I know that, but as long as we stay on subjects of techniques, I tend to get work done there. But really the amount of time you invest beforehand in really finding out what people want from their samples and what they're interested in can pay off a lot in the end.
Thank you. Do you have any thoughts on that, Eilike?
Yeah, so I know there is a fascination for cryo workflows, and they are fascinating. And just to say that there is plenty of questions that you can answer without using cryogenic conditions. And it's something that we discuss very often. We understand the temptation to be ready for everything, but let's just cross and first answer the question that you immediately want. And if it's worth it, yes, definitely we should embark. But cryogenic workflow gives you a lot of information, and it's a lot to dig in. So we have to be careful before embracing any path, and having both works pretty well. So
I'll make another comment to that. My goal when someone comes up in a project to me is to de-risk. And de-risking could also mean using light microscopy as one of the tools to answer their question, or at least to triangulate what it is I need to get for them without screening a bunch of material that is very expensive. The worst thing in the world I can do for a PI is to come in with a project that I'm taking so many risks on. So that's part of the reason why I have a protocol that is generally robust. But oftentimes I steer them to light microscopy to make sure that I'm getting exactly the target they want there and understanding it before I proceed to the volume. And then, of course, one last comment that must be said in this room. You put a lot of metal into a sample if you're talking about plastic. Cryo is another thing, but you don't know where your sample is sometimes in thick, bulky materials. And so I try to use X-ray again to keep de-risking the project. So I'm not taking thousands of useless sections and sending them a large bill or absorbing it as a cost of development from my own side.
Lovely, thank you. So from that, there seems to be that you're spending a large focus of your time making sure that you're directing the samples in the correct way. How do you get the scientists involved in that sort of pre-screening and pre-filtering of what they need? Are you providing them all the information, or do you have requirements that they cannot come to you for an experiment without having done some initial background work, so light microscopy, or have an idea of what they're actually wanting?
So yeah, definitely. Again, we're going to high resolution techniques. So any information, as you mentioned, with present microscopy or any previous work is going to help us simply to target what we should look for. So coming with preliminary data is always a first, simply because you're going to facilitate your life, and that is what we want at the end of the day. It's something that we do tend to expect some good question that fits well the technique that you want to use.
I'll make one other comment, and I think it was kind of alluded to already, and I think perhaps a lot of you can identify it. The PI comes up to you and slaps a paper on your desk and says, I want you to reproduce the work. But you look at the paper and there are like 5 sentences of how they did the protocol. And so part of your job is to be a detective and, again, take that and extrapolate. And by the way, how many times has someone slapped a piece of paper on there and said, oh, I think I can do that a little better? So my comment is that you may have to refine it a teeny bit as you go forward, but I think it still comes down to the crux is what is the scientific question that has to be asked. And I think everybody, if we do our homework up front, we have less chance of disappointment. However, I would say about 80% of the time, once you've done the volume, they're like all excited because there's 25 things they didn't think about. And so now you also have to keep the mission creep from going.
Lovely, thank you. I think that sort of touches on, how are you able to take information from a published protocol and adapt it? I mean, like you said, multiple times you find that it's 2 lines of we performed electron microscopy on sections, and then trying to interpret how that was prepared. So are there any approaches that you can think of that we could as a community start trying to adopt to be able to move forward to having clear, unambiguous protocols that everyone can understand and interpret? Maybe Rob, you seem to react to that.
Yeah, well, personally, I like, for instance, what we have now is that you have these databases where people submit their paper already before they actually publish it. And often the nice thing about that is that the materials and methods section is at least 4 pages long. Because the only restrictions there are the restrictions imposed to us by the people that actually own the journal saying this should be 3 pages because otherwise we lose the interest of the readers. So often these pre-publication submissions, they actually have the good materials and methods still containing all the teensy bits of information that you actually need. The other way around it would be to, let's say, create something like that to which people can post their protocols, but then preferably also expanded protocols. Say, okay, we use this and this to bring these and these types of tissue cells, whatever. In our hands, it works. This is the list of chemicals we used, also stating specifically what type, because that can really change a lot. And then it would be much easier. But of course, this is a lot of effort for everybody to do, but it could really change the world. On the other hand, I know that some people would prefer to have their own protocols kept to themselves because the chance of being scooped, of course, are also big.
You, Rob, Michelle is dying to get in there.
just on that topic, so we have something called a sample preparation widget that has been worked on for quite a while now in between the EMBL EDI and the Rosalind Franklin Institute. And the lovely Nick, who's sitting just here, if you want to raise your hand, will be presenting a poster on it and doing demos on Thursday. And it essentially fills that niche exactly. So come check it out.
Thank you very much. Yes, of course.
Also, what I would suggest is, as a reviewer or even people talking to each other or publishing, if we can put success rate when we present new techniques, it's very nice to say, oh, I've got these beautiful techniques, but it works 1 time over 20. It's great, but it's 1 time over 20. So it would be good to begin to pay attention to success rate at any step.
I would encourage you as a reviewer to demand that the authors of the paper put in the proper details. I would also go a step further in making sure, and in addition to the sample prep widget, which if you didn't hear about it today, please go and stop by and see that, because this is an opportunity. Deposit your data into a shared resource. Provide the metadata in which your data sample was collected under, because that's the other thing. When people get a certain contrast, it's the details of the protocol, but it's actually how they image it. And that's usually the least information. It's the voltage in the model of the instrument, and that's about it. And then, but it all matters. We can set our own examples to the community by doing it ourselves, but also to make sure we request it when we're reviewing papers as well. It's a very simple ask. When you do it, and people use it like that as an easy win that they go on.
Lovely, thank you very much. Oh, Matt.
Thanks. I mean, I think an important point about this is that you have to understand the way that people think and write these protocols, because very often when you're describing to somebody how you do something, you forget all the things that is in your mind all the time that you don't need to say. And this is how we end up with like 3-line protocols. And we miss all of the kind of steps that we do in between the imaging and the sample prep, just cutting something and mounting it on a pin. So I think we have to think about that sort of thing when we're writing these protocols as well.
Thank you. So we also have another question at the front as well.
Now it's working. Sorry. Yes, maybe the electron microscopy community, they have to do something similar what we do in the light microscopy community with the quality control. They have different working groups, and one of them is actually for what do you want. They have to be necessary in the protocols, not only for the sample prep, but also for the microscope in the journals. Believe it or not, the journals have nothing. And they really have the minimum, want the minimum. Maybe we need the community find this minimum. They have to be there, but they have to be the EN community where actually come up what is the important points to have there. You will never have 20 pages for material methods. It will not happen. Of course, you can use protocols.io or other systems where you can link your protocol with all the details, but maybe the community had to try to find some points that will be interesting, that they will be there, that they will make reproducible every material methods.
Very much. Before I go to you, Matt, so that to me sounds like you're suggesting maybe adopting a minimum reporting guideline type approach. I know it's commonly used within the proteomics realm. So something like that across the EM community might be quite a nice approach. Thank you. Sorry, I think Matt had his hand up again. Oh, sorry, this is in the middle, sorry.
I'm curious how you all approach this. In our facility, we have started insisting that when data gets published, they come to us to ask for us to help them write the protocols for everything. And in fact, we've stopped giving them this information so that they have to come to us when it comes time to publish. And then we write it for them, and then we ask to proof it when it goes through the journal as well. And inevitably, we're always finding mistakes and we're fixing them and adding them, but this way we can ensure it gets added. I'm just sort of curious if other places have done this kind of stuff.
Well, let's say for me, every time I do something for somebody, I indeed write the materials and methods myself and say, okay, this is what I did. If you want to have it properly, then copy this, put it in the slide spots, and continue. But I don't have any, so most of the collaborations I have are people that keep on returning to me using comparable samples. So for me, that's relatively easy because I just have to copy and paste and slightly modify. Yeah, that's about it.
Yeah, I'm in a similar position as well, that we haven't had people try to reduce or modify any protocols and material methods sections that we have sent them. They have just taken it verbatim and put it into the journal. So we've been, I suppose, very lucky in that respect. But I don't know if anyone else has any steps to prevent this happening, or any thoughts on how you could improve their ability to take your information and use it correctly.
We don't have that policy, but it happens by default because they just let us do most of it, so they just don't know. I would go another step further, that when you develop that habit, they'll also come to you when they're writing the proposals. And so nothing like being a reviewer and reading some technical, sophisticated volume M proposal, and the PI didn't really ask the expert on the right language or resources, and you feel you don't have confidence they can achieve that goal. So it's actually both sides of the equation. One is the downstream when they're funded, and the other one is the upstream to make sure you build that relationship with them up front, and then they trust you, and then they get successful funding.
Lovely, thank you. And Matt, do you still have a question?
So it was a kind of response to the comment about minimum reporting. So there's a couple of papers about that. One is a couple of years ago, which was REMBI, I think, REMBI. And that was an exercise that I was slightly involved in of trying to have a suite of minimum metadata that you would associate with a publication. And it's difficult because people have lots of different opinions. And of course, if you're talking about lots of different workflows, then the minimum metadata might be different for all of those. So having a kind of universal one is challenging. There was also another paper where Chris Petty from the CRIC talked about minimum metadata for reporting serial block-based data. Since that came out, I've certainly seen publications where that minimum metadata was not there. So there are resources out there for you to kind of use to do some of this stuff, but it is a challenge.
So maybe that's more of a community approach, that we should all try to adopt something that enables us to be able to have reproducible and consistent preparation protocols. So maybe moving on, I think probably for me, one of the biggest issues I have is trying to get consistency across sample preparation, either from samples that have been sent to me by different labs for the same sample, or when I've been moving from facility to facility. Do you have any approaches on how you try to maintain consistency within your sample preparation, or any ideas on what you think the biggest barrier is to enable this consistency?
Main problem I see, especially if I got samples sent to me, is how they prepared their sample before sending. So the workaround I was told there was, if people want send their sample to you and it needs to be prefixed, please send them your fixative first. And then they use that, and then they send the sample back. And they should use the protocol you provide for that and not think of something themselves. Because that was the only way for us to, at some point, get consistent results using prefix material. And the other thing was, before I went from Utrecht, where I did my PhD, to Nijmegen, first I had a very long chat with the main technician there, just checking, okay, what are we actually using? Which chemicals come from where? What is your experience using different types of different brands or different grades? And just making a list for myself, saying, okay, this is what we have been using. So starting with new job, and we make sure that if I wanted things to be reproducible, I should at least have this. And I've been doing some checking out, and actually I found out as long as I stick to spectroscopy grades in terms of solvents or other things, then often it is reproducible enough. And at least you can get, with the spectroscopy grades, you can get these data sheets where they actually analyzed what is in the specific batch. But in case you find inconsistencies, you can check back, okay, did I use a different batch? And then pull up the data sheet from those batches and see if there was actually something wrong there or whether you just screwed up somehow.
Lovely, thank you. So I would like to say that we're not a facility, so we don't receive sample from outside, but we do completely accept on any beginning of a project that we plan for manpower and money for solid testing before we enter into the pipeline. So very often a project is going to take 2 to 3 months to stabilize its sample preparation. Hence the point that I made earlier on measuring success rate. So every project would start with measuring on 3 independent experiments the success rate of a given procedures. And it takes time and it takes money, but once you have it, you're good for it for the rest of the project. And very often you're going to see that you have a good transfer to another project because they're close enough and relative enough that you have minimal change to do. So there is a cost to it, but it's a cost that has been for us totally worth it.
I'll make one other just quick comment. The first step, in my view, and I think it relates to your comment, is the primary fixation. It's that first fixation. And if you blow that, nothing you can do is going to recover it. So one thing I typically do is I move and I look into the literature for their particular organism if it's one I haven't dealt with before. And is the stuff in the literature high enough quality? And if it's not, then we might start inventing. But in the meantime, it's this first fix. Surprisingly, you've had maybe a technician or yourself, somehow something comes up and you can't get your protocol in exactly the 3-hour slots that you said you want to, and sometimes it sits over the weekend, and then you find out, well, some of the downstream stuff is more tolerant. But it's that first fix is where I usually put my energy.
Lovely, thank you. And Rob, there was something that you touched on there that quite interested me, that you were talking about how you were recording every single step in every specific detail. Is there any way that you do this? Do you have specific software? Do you have a spreadsheet? How do you keep track of all the possible variables that go into your experiment and then sort of monitor the outcomes of them?
Well, the first time I actually went into this was when something just didn't work all of a sudden. And I didn't have all this backup data. So mostly I just work with a spreadsheet, but it's mostly for reagents, solvents, and other things where I just keep track of, okay, what do I have, where it's from, and what grade of quality do they keep to it. Because for most, especially for the spectroscopy grades, you can actually put in a batch number and then get, for instance, the data sheet for that particular batch number. Depends a bit on suppliers, and there you're also limited in which suppliers are actually available in your institute or in your country. And so that is also a bit tricky in terms of getting universal protocols that work everywhere. If you can't get to that supplier or you can't get to that person or reagent, then yeah, nothing that you can do except for try the best with whatever is available.
Lovely, thank you. Do either of you have any amazing solution to this problem that we all face?
So my team actually uses OneNote if you have Microsoft. And so we have all of our protocols based on date and listed, and all the reagents that are used and the part numbers and so forth. And so when we see a deviation of something we've run before, we can quickly go in and look at what's different if we change the reagent recently. But that's the only way we can track it, because if it's in your head, which a lot of people out of habit just say, oh, I know that protocol and I'm just doing the same thing. We actually keep notes on it. And it's easy once it's in there because 95% is already written. You just would go modify it.
Lovely, thank you. I can see Michelle is twitching again. So.
I'm like right in your line of sight. So this is a broad question, maybe for the panelists, but also for the room. Are there any like electronic record notebooks or electronic lab notebooks that anyone has used successfully in like the volume EM space?
Let's say in our lab, at some point we were suggested to use benchling. When I had a look at it, I thought, this is not what I want, because that was so extremely aimed at doing genetics, genetic modification, noting down which factor used for what. Like, I use up all my free space if I just put in 3 EM images. So I've only used it to store protocols for now, because it's an easy step to put in the protocols. But then again, I also liked very much the setup I had for my protocols in Excel, which was easier to just type in, change the number, and everything just works out because I used the formulas to calculate how much of what I needed everywhere. So I know there are a few decent systems on there, but often they lack the possibility to link to image files, for instance. So the only way around it I've seen so far was actually then using a spreadsheet and then having a cell that links to a particular folder. But the minute you move the spreadsheet or the folder, everything's screwed up again. So unfortunately, I have not seen anything that works nicely.
Lovely, thank you. So Matt.
So I've looked at a lot of these, and there's a lot of very powerful stuff out there, and a lot of it is pretty overwhelming. I asked about this in the Slack workspace a few days ago, and we had a couple of responses from people who had created their own tools for kind of their own personal use, and they were happy to kind of share them. And there was definitely a feeling amongst the discussion that these things are overwhelming, and we just want something that's fairly simple, that we can either track samples or track metadata, and the software that I've used, like IMS, where you can have a database of images and have all of your metadata associated with that. The amount of implementation that's required, you'd need a couple of staff members to actually get it to work how you want, and even then, there's a lot of user input. I mean, maybe guys from the CRIC can comment on this, because I think they've taken it further than we have done at King's. I'd be really excited if somebody had a good solution to this, but I'm not sure a simple one exists at the moment.
Okay, thank you. John?
Can I just pick up on something that Kirk said before about sometimes out of our control, you get times when you increase or decrease your incubation times just out of circumstance. It would be nice to have, when we record our protocols, some kind of idea of tolerances of time, because I'm a stickler for sticking to a protocol, and it has to be a 10-minute incubation for increasing ethanol concentrations or something like that. But occasionally, they don't always have to be so strict. For instance, the other day I was doing a serial block-based protocol from the CRIX protocol for that, but then I decreased increasing Dirkpan resin concentrations from 2-hour incubations down to 1.5 hours, because I didn't have time in the day to get it finished in time, and it worked fine. Maybe by fluke, but it'd be nice to know from people's experience on a recordable database just how low you can take it, how you can take it, maybe in concentrations of different reagents as well, but in times for incubations would be invaluable, I think.
I think that's highly dependent on what type of sample you have. I mean, if you work in monolayers, you're extremely flexible, because monolayers is just one layer of cells. Also dehydration, 2 minutes for each step, if you change twice, would be good enough to actually get decent infiltration. unless you're working with monolayers of fungi, for instance. That becomes more tricky. So really the main thing there is what is your sample volume? What is the viscosity of the stuff you're trying to get in, which determines time. And so there is, if you think a bit of, okay, how big is this sample? Are there any weird barriers in there in terms of cell walls or highly glycosylated layers or whatever that can influence penetration or dehydration? They can often see quite easily, okay, this is, here I can actually tweak it and here I can increase the limits a bit in what time I need. And for other things, then there are a few steps that have been recorded in literature where you can say, okay, if I'm not going to make it all the way, for instance, in dehydration, then stopping at 70% solvent is often a decent step where you don't have too much influence later on, or stopping at the first resin step and then taking that overnight often also works. But whatever you do, don't stop at 100% or don't stop at, let's say, 30% in terms of, because those are the ones that actually start mattering in terms of either swelling or actually shrinkage. So yeah, the rest is either trying it out, see it happens, or really thinking about, okay, what type of sample do I have? How big is it? Are there any weird things that I know would be happening with this sample?
Thank you very much. I must admit that on my protocols, when I've been developing them, I have taken sort of liberties with some of them. So I know that maybe I don't have time to finish it and I will put something in the fridge overnight at 4 degrees. And if I don't see any downstream effects, then I note that in my protocol and have, here's a break point, you can put it in the fridge. To enable people to be able to design their days around protocols that sometimes can last sort of 10, 12 hours. So I suppose probably moving on, what would you say is maybe the one gap in the sample preparation landscape that you would like to see filled?
Okay, so for me, it would be for creative workflows. I think we need to have a good understanding of every single step of these creative workflows and how impact the previous imaging to the second one, and how the first imaging technique or the first step of your work, your creative workflows, is going to impact the media or the environment around what you've got. I think is missing, or the beam damage or all of that. So for me, the correlative workflow needs a lot of digging on this one.
If I could wave a magic wand, I think I would consider sample prep in the framework of my downstream processing, meaning the biggest amount of energy and work actually goes in and segmenting out and quantifying and measuring the structure you want to. So what I'd really like is a list of protocols that will enhance microtubules. And then it was easier for me to segment than going in and doing it on tracking. You know, there's good ones out there for mitochondria, kind of, sort of, right? It's the membrane and stuff. And you can enhance certain structures, but I actually would like them to be, you know, start playing with chemistry to target some specific structures exactly, so it makes segmentation downstream later easier.
I think linking on that, getting chemists involved in electron microscopy would be brilliant. We have all these things that we don't actually know how they work, or have a slight hint of knowing how they work, or just based on pure guesstimation how they work. Well, if we can actually get a bit more background into what is actually happening there, it's much easier to predict, if I change this, then this will happen, leading to that. And really, until now, I've always had to dig back into the 1960s literature to get a decent explanation of what is actually going on during, for instance, chemical fixation or osmium fixation, or what the effect is of adding potassium ferricyanide or ferric cyanide to your osmium. And if you change that, doesn't matter. All these things is, well, parts of it you can find back in ancient literature as soon as you can actually access the ancient literature. Because it's way too often I run into the issue that I think, okay, I finally found the article I'm looking for, and then it's nowhere to be found. You see an abstract, you see 60 citations. But then I'm wondering, where do all these people have this citation from? Did you actually read the article? Because I can't find it. But it's a bit my personal rant.
Oh, lovely, thank you. We have a question from the front.
This is more like a comment. So currently, I think that in the lab, everything is going into the kits. And with kind of younger generation of students, they don't even know how to make a phosphate buffer or almost like any buffer. So I think that the discrepancy between the EM understanding, because we really need to understand every step and exactly going, understanding if you want to contrast membranes or some cytoskeleton or ribosomes, you have a different protocols, which means that we are definitely not working like as a kit or like single protocol, and there I think that's one of the challenges, that how to engage the users to kind of really appreciate every step.
Lovely, thank you. Oh, I see Matt has a comment to that.
I mean, this is something that we've talked about on Slack quite a bit, and Rob has been fantastic at explaining some of these older papers to us. It's also an exercise that we went through as part of I think the sample prep working group, to compare a few different serial block-based protocols, and that was a very illuminating exercise, particularly with reference to kind of, how long is this particular step? Well, it actually varies from person to person and from lab to lab. I almost feel like, and part of that exercise was to look at each step and say, how much do we understand about what this is doing? And I'm sure there's lots of kind of great books and so on out there, but I almost wonder if it's something that the volume EM community can organize as a database of, this step, this is all that we know about this one step. And searching, now that we have access to all of the history in the Slack workspace, we can pull a lot of that information together, but it's going to take quite a lot of work to collate that stuff. But it could be really, really useful.
So real quick, you've now mentioned the Slack workspace 3 times. So do you want to give a broad overview of what it is for anyone who isn't aware?
Sure. So this is a Slack workspace that I set up during the lockdowns a couple of years ago, and it's called EM of Cells, Tissues and Organisms, because it's kind of a broad biological electron microscopy discussion group, and it's for anybody who's involved in electron microscopy, from students, researchers, academics, vendors, and anybody else who I might have missed. At the moment, we are, and we've partnered with the Volume EM initiative, so we have a Volume EM channel, but we also have various channels for kind of broad topics like sample prep, imaging, image analysis. We've kind of resisted going more granular than that, because it kind of reduces the quality of the discussions. But so yeah, that's kind of what it's about. People can go to the EM of CTO Twitter account. So if you just type it like that, and there's a sign-up link there, and there's going to be an announcement about kind of funding in the next couple of days as well. So. Yeah, that's where we're at. But anybody can join, and you're welcome. And if you have any problems joining, email me or get in touch with somebody who's already part of it. Thanks, Michelle.
Lovely, thank you, Matt. And yeah, if you aren't on it, please go and visit it. It's absolutely fantastic. John? Yeah,
I just thought this is a good point to mention about, I know 90% of people might be already signed up, but to the Tech EM seminars, we're coming up now, the ones that are run by Erin Tranfield online. Contact her in Ghent and to sign up. But in the coming months, there are going to be pure one reagent discussions to work on things like osmium or fixative. I think we had a whole impromptu discussion for about an hour, 2 hours on which fixatives to use for certain samples. So I think this is a good forum to get that kind of information. if people could then record that and collate it and put it onto a database, that'd be even better. But I'd encourage anyone to sign up that hasn't already signed up.
Lovely, thank you. And oh, there's a question in the front, sorry.
Just behind you.
A question about the uranyl acetate. It's not allowed to buy. I don't know if here also, but we are still using it. So what is your suggestion for some new contrasting agent, or if you can comment it?
Lovely, thank you. Has anyone got any experience on using some of the UA-less or new lanthanide type agents?
No, but I'm trying to find time to test one or two.
Yes.
But this is mostly trying to find time too. So for me, having UA is not that much of an issue since I inherited this very old bottle of mercuronyl acetate. And then I inherited another bottle of mercuronyl acetate, which was completely broken and duct taped together. So I very gently, under very nice conditions, poured everything together, closed it off, and never told the AMD that I actually did that. So personally, I think that some of the restraints, the uranyl acetate, are a teensy bit overrated, considering the amount of radioactivity it actually has, compared to, for instance, a fresh gypsum board. If you put your Geiger counter next to fresh gypsum board, you'd be surprised how much background radiation that one gives. But of course, this is long-term, so you need to dispose of it as being long-term radioactive waste. And of course, it's a very heavy metal, so in that circumstance, you should not eat or drink it or touch it with your bare hands. But this will also be the same for lanthanides, because they will also be heavy metals, so same holds there. I think the main trick with uranyl acetate is that if you look at it, it is a relatively, except for the fact that it's really heavy, it's a relatively small molecule. Also, if you compare to all the other lanthanide groups, they tend to be much more bulky. So in terms of grain size that you end up with, other lanthanides will be also more bulky. So you'll lose a bit of resolution there. And it's really, I've seen some very good results and also seen some very bad results. I know that Erin Cranfield, when she still was in Spain, actually had this data set of things they tested with different uranyl substitutes compared to uranyl acetate in terms for post-staining. Yeah, there was one there where I thought, okay, this looks decent, but not as good as I would get with uranyl acetate. So yeah, I think it's tricky, and you really need to test, and sometimes actually combinations of different lanthanides might actually work better. But then again, if we have more understanding of how these things work for the other lanthanides, because for uranyl acetate, there's a decent understanding what it binds to and what it does. But for the other lanthanides, we don't know. Nobody's tested it.
I've used the uranyless, for example, just on sections post-staining. And this actually is probably a reflection of my age. It was a little less satisfying than uranium was. Other than that, it would be fine for a lot of purposes. It depends on the question. What I don't know is how would that affect the volume EM run? Has anybody used any of these for a volume EM run? Because if no one has, I will go home and do it. It's just side by side. Go ahead.
We do have some hands.
So we use neodymium for our BEM and our routine TM stuff now, rather than using UA. We do it in an NHS setting, so it's been quite rigorously validated against our patient samples. But it works really well with volume EM stuff as well, the serial block test.
Lovely, thank you. Got Ian as well.
So I think what's quite interesting is when we're talking about sample prep as well, we saw Ria's talk earlier where she was talking about removing some of those heavy metals. I know we've done that occasionally, though, mostly through mistake, where naming no names, but out of the two of us, it wasn't me that altered our protocol by just forgetting one stage. We do that sometimes, and actually, what's quite interesting that Ria's results were absolutely fine, and as we see industrial partners, improving their equipment and the sensitivity of their detectors, it may be that coming into the future, rather than spend all our time now worrying about, because Kirsty, it's just you. With us, it's just the two of us. We don't have all the time in the world to meticulously change
each step of what is a very multi-stage protocol to get to our final samples for VEM, it's actually going to be our industrial partners that save us by producing, you know, faster, more sensitive, better detectors, so we can actually start reducing those heavy metals. And that can be, you know, that's more likely to happen before I run out of UA or before Rob runs out of UA, by the sound of it. So this may be that it's not a tree we have to bark up, if I mix metaphors a bit there. We don't need to be pursuing that necessarily. It might be that if we all actually experimented in reducing our heavy metals anyway, then we'd still be getting good data, as Ria displayed earlier.
Thank you, Ian. Okay, so I think maybe looking for the take-home messages from this session is that we should all start investigating the Volume EM sample prep widget and trying to populate it with some of our protocols. We should potentially all be looking towards making sure that we have information about reproducibility and success rates on our sample preparation. And we should maybe, as a community, start looking towards trying to work out how we can ensure that we have consistency and standardization across reporting of our sample prep protocols. So I suppose one of those sort of key things is there's a lot of information there, and it's a lot of work for people to start putting it into documents. So how would the community and how would this panel like to approach the sort of the transfer of knowledge, whether it's from the tiny tips and tricks on which particular hair you want to use for your loop for manipulating your sections down to guinea pig is very good. Down to, you know, fixation duration, buffer formulations. What would you say is going to be the best way, in your opinion, to do that, Rob?
Can I get 5 minutes to think about
it? Of course, if anyone has.
I'll respond quickly, and this will be another plug because we have the Chan Zuckerberg Initiative funded a video kit. And to be honest with you, if you have your little sound bite and you can spend, you know, 20 minutes or something to put something together for the way you do it, I think our library will quadruple probably in the next several months. But we're looking, the kits are available, you know, take a look at it if you're willing to do it and you have somebody, some idea, a little technique that we're talking about and how you manipulate your sample or prepare it. That's one way that we can help and share it more broadly, where sometimes a written word in a text form is not enough. We have an opportunity to train people almost as if you're there personally.
Lovely, thank you. So Alice at the back.
So I think this question is open to everyone, to maybe the community for Volume EM, you know, Joe mentioned about the follow question you raised regarding the chemistry of each of the people we use in the literature. Everyone has to figure out what is the account. As a community, if it's possible, I think it's better if we have a typical good reference to list, right? Typical principle and also good application on that. Then that will save a lot of time for the young people, and it's hard for us. Maybe it's easy to dig into all the literature, but for the student or young people, maybe it's a big pool for them to try to fish out some of the periods.
If I might just say something about that. As much as I love database and everything, what makes sometimes database not useful is that they're not searchable, or if we don't know how to ask the question to the database. And I know that we're trying to do our best, and I know that Google and AI is going to serve the world. But in the meantime, we need to find ways to put a database that is searchable with keywords. And the problem with keywords is that you need to know the keywords to search for them. And so sometimes I see myself asking the question to a colleague rather than reading literature, simply because my colleague is intelligent and will understand the meaning of what I'm trying to say beyond what I'm saying. And so I think is where the challenge will remain, is searchables and being able to ask questions like, what is the best protocol for microtubule labeling? But why is best? That is the first question. Where do you put best? Is that resolution? Is that contrast? So this type of question that we still have to answer to.
I'm just going to jump in ahead of Rob there. So to me, that sounds like we maybe need to have a common language across the whole of the sample preparation protocol. And we should maybe start looking towards trying to make sure that when I say I am using this as a fixative, that everyone understands exactly what I mean by fixative. And maybe this should also start to be incorporated into our reporting, that we are all using a commonality of descriptors. Sorry, thank you. I'll let Rob.
Yeah, but then of course you end up with the semantics of what is being a fixative, because then I know at least 6 people who start a rant on that osmium is not so much a fixative as that it just completely destroys everything it touches and oxidizes everything. But the same can be, for instance, said for potassium permanganate, which is, well, if you look through literature, is often cited on one hand saying it works brilliantly, and on the other hand saying it completely destroyed all the proteins in my sample. So yeah, we may have to find some common ground somewhere in between there.
Yes, some consensus agreement across the community on what we actually mean when we use a specific word. So I think approaches are being taken into that within the sample prep working group, and hopefully there will be more information coming on that and how you can participate in that in future meetings. But in the meantime, if anybody else has any further pressing questions for the sample prep panel, I'll take them. Alex?
I'll make one other comment. So I'm going to, again, protocols or sample prep widget, we have multiple tools out there, and I want to speak in benefits language because there's a barrier for you to go in and assemble all that information and keeping it updated and so forth. However, I want you to think about the benefit. If all of your colleagues are doing it, then you can get something out. But if you're the only one doing it, so please participate. Let's show the value of it. And if you get a couple wins and say, oh, this was useful to me, then others get them on board. And I think we just have to propagate it.
Lovely, thank you very much. So that has reached the end of our time for the discussion. I want to thank all the members of the panel for the valuable input. And I would like to encourage you all to check out the Volume EM websites, the Slack channel for Matt and Erin's Tech EM seminars because they are invaluable sources of information for sample preparation.